How to hack your cells to cure cancer. The future of medicine? The first known ancestors of humans have known to exist for the past 6 million years, and the modern man about 200,000 years ago. The entire human body is composed of multiple organs and tissues, which in turn are a group of individual cells. These cells have been programmed to perform only a single task: to preserve the genetic code within the DNA, and duplicate it to pass on the code to the next generation. This duplication is something that's not within the control of these cells, and thus leads to different varieties of copies of the genetic code. Sometimes these newly created genetic code variants can make a cell divide uncontrollably, leading to uncontrollable growth, eventually multiplying itself into a tumor, which we describe as cancer. Since its discovery, treating cancer has been a monumental challenge. Despite decades of study and billions of dollars invested in clinical trials, we still have a problem with cancer medicine delivery. We still provide chemotherapy to patients, which is so non-specific that although it kills cancer cells, it also kills the rest of your body. Yes, more specific medications have been created, but getting them into the tumor remains a struggle. They end up collecting in other organs or passing via your urine, which is a complete waste. This is where the biohackers, a new breed of synthetic biologists, enter the story. These biohackers are rewriting the code and fixing the unfixable by combining science, medicine, and engineering. Biohackers reprogram a patient's immune system to spot and eliminate cancer cells by hacking into their genetic code. CAR T-cell therapy is what it's called. The immune system, which is primarily made up of white blood cells, protects the body from hazardous microbial invaders. T cells and B cells are the two types of these guards. T cells use special claws, which are antigen-specific receptors, to check those antigen IDs. If they locate a match, they attach and release deadly chemicals that cause the invader's membrane to burst open. Their B cell colleagues make antibodies, which are tiny proteins that attach to antigens like little claws, designating them for destruction. These two guard cells, along with your immune system, are exceptional in detecting and combating diseases that enter from the outside. They're not so effective at detecting renegade cells in your own body though. Cancerous cells have antigens that look a lot like your own cells, and your T cells and B cells aren't prepared to attack them. The standard cancer treatment is to remove the tumor or use radiotherapy and then chemotherapy to destroy or slow the growth of cancer cells. But you can't do that if it's a blood cancer that's spread throughout your body. And if the blood cancer originates in your white blood cells, your immune system's essential guardians, you'll have a hard time detecting it. This is the situation with acute lymphoblastic leukemia, and CAR T-cell treatment makes a difference. The biohackers retrain a patient's immune system to detect specific antigens, protein fragments on cancer cells. You'll need millions of a patient's T cells to get started. Then to have a T cell perform something else, you must replace its default code with a custom-made one. Modeling a new genetic code is precisely what biohackers do. 3 crucial instructions are included in the new T cell code. Number 1. The code instructs the T cells on how to identify and kill cancer cells, how to alter an antibody, or what B cells do to grab onto a target antigen. The antibody is changed to create a novel receptor that can detect the antigens found on the tumor. When it detects that cancer cell, it is told to generate copies of itself. It tells it to stay alive in the body of the patient. You utilize a vector to deliver this new code into the patient's T cells. It's something that can readily infect the T cell and transport the customized DNA in within it. The CAR T cell is then formed. The term CAR T cell is derived from a Greek fire-breathing monster with a lion's head, a goat's body, and a serpent's tail. It was given the term CHIMERA, which has now come to refer to anything that contains 2 or more types of tissues or cells. It's a CHIMERA because the genetic code of this newly created cell is a half T cell, part antibody, and it goes in quest of cancer's antigen via its new R receptor. You give the patient a moderate dosage of chemotherapy to wipe out their current T cells before reintroducing the amplified T cells. Then you simply reintroduce the CAR T cells, which continue to migrate and search according to their usual DNA programming. They've changed what they're looking for owing to their new code. They're now on a mission to identify and destroy malignant cells. CAR T cells are live medications that stay in the patient's bloodstream for years, unlike traditional chemical-based drugs that are quickly used up or eliminated. On the other hand, they are costly. Each CAR T cell treatment is tailored to the patient. Getting them to function with common diseases like breast cancer or lung cancer is more challenging since the CAR T cell needs to target a specific antigen on the cancer cells, which is much easier to detect in blood malignancies. But it's still early days, and CAR T cell therapy has a bright future ahead of it. Researchers like Dr. Martin Puel and his team at the University College of London aim to improve leukemia and lymphoma treatments even further, and solid cancer research has recently shown promise. The survival rate for B acute lymphoblastic leukemia has improved dramatically thanks to CAR T cell therapy. Virtually all patients now go into remission, which means the illness is no longer detectable. Biohacking is the new paradigm in oncology, and it can reprogram the genetic code in your cells to enable your human body to do things it's never been able to do before.